The pentose phosphate pathway is a cytosolic pathway that interfaces with glycolysis. What's important to note is that in this pathway, no ATP is directly produced from the oxidation of glucose 6-phosphate. Remember, glucose comes into the cell and is phosphorylated to glucose 6-phosphate. Up until now, we've seen glucose 6-phosphate being oxidized through glycolysis or being stored as glycogen. And the pentose phosphate pathway is an alternative metabolic route. So the key features of this pathway are illustrated here. This pathway is important for the production of NADPH. Second, it also produces ribose 5-phosphate, which is our 5-carbon sugar used for DNA and nucleotide synthesis. And finally, you can see here that the intermediates of the pentose phosphate pathway can be regenerated back into glycolytic intermediates. And those intermediates will be fructose 6-phosphate or glyceraldehyde 3-phosphate. So there are two parts of the pathway which are distinct and can be regulated independently. The first phase is the oxidative phase. This consists of two irreversible oxidations that produce NADPH. As noted, NADPH is required for reductive detoxification and for fatty acid synthesis. The non-oxidative part of the pathway allows for the conversion of ribulose 5-phosphate into ribose 5-phosphate. The big take-home here from this non-oxidative part of the pathway is that all of these interconversions in the non-oxidative pathway are reversible and use the enzymes transketolase and transaldolase. These two enzymes move 2 or 3 carbon units onto other sugar moieties to generate a variety of sugar intermediates until we result at glyceraldehyde 3-phosphate or fructose 6-phosphate, which again interface directly with the glycolytic pathway. So there is a single key regulatory enzyme for this pathway, and that enzyme is here, glucose 6-phosphate dehydrogenase. This enzyme oxidizes glucose 6-phosphate to 6-phosphoglucolactone, and this compound subsequently undergoes an additional oxidation and decarboxylation, resulting in ribulose 5-phosphate. Glucose 6-phosphate dehydrogenase is regulated negatively by NADPH. High levels of NADPH will inhibit the activity of this enzyme, and therefore NADPH is only generated when the cell needs this. The non-oxidative phase, our reactions down here, are not regulated, and in conditions where there is high demand for our 5-carbon sugar, ribose 5-phosphate, for nucleotide synthesis, the non-oxidative part of the pathway can function independent of the oxidative phase to produce ribose 5-phosphate from our glycolytic intermediates, glyceraldehyde 3-phosphate and fructose 6-phosphate. Again, the key take-homes from the non-oxidative phase of this pathway are the enzyme transaldolase, which transfers 3 carbon units, and transketolase, which transfers 2 carbon units. This enzyme also requires thiamine pyrophosphate as a cofactor. The final piece I want to draw your attention to in this figure is the reaction catalyzed here by PRPP synthetase. We'll look at this reaction when we address nucleotide synthesis, but this reaction converting ribose 5-phosphate to phosphoribosyl pyrophosphate will be our starting point for nucleotide synthesis. So we've talked a lot about NADH in the course. Let's take a moment to look at the role of NADPH. So first, NADPH is a high-energy compound used for reductive biosynthesis, as it cannot be oxidized in the electron transport chain. It's also used by many tissues to scavenge and detoxify reactive oxygen species, which can cause cellular damage. So glutathione is a tripeptide compound consisting of glutamate, cysteine, and glycine, and it plays a key role in scavenging reactive oxygen species, again, which can cause damage to both DNA and cellular proteins. The connection between NADPH and glutathione is illustrated in this figure here. NADPH using glutathione reductase is used to reduce glutathione and retain glutathione in its reduced form. Reduced glutathione is then available to detoxify peroxides. And of important note, the reduction of glutathione in the red blood cell is done exclusively through a series of oxidation reduction reactions using NADPH. So we've seen how the pentose phosphate pathway interfaces with glycolysis, is able to produce NADPH, as well as 5-carbon sugars required for nucleotide synthesis. So let's ask the question, what is the role of the pentose phosphate pathway in the red blood cell? So there's a couple key points in the red blood cell to be aware of. First, red blood cells, in their mature form, lack mitochondria. So there's only cytosolic metabolism going on. This means that glucose coming into the red blood cell is phosphorylated to glucose 6-phosphate and can either be oxidized through the glycolytic pathway or enter into the pentose phosphate pathway. The pentose phosphate pathway plays a very important role in the red blood cell. In red blood cells, this pathway is the sole source of NADPH for the reduction of oxidized glutathione. Again, the reduction of glutathione, as we saw on the previous slide, removes hydrogen peroxide in a reaction catalyzed by glutathione peroxidase. And finally, this reaction is important since the accumulation of hydrogen peroxide decreases the lifespan of the erythrocyte by causing oxidative damage. In other tissues, NADPH, we will see, can be generated by the cytosolic enzyme malic enzyme. However, this enzyme is not present in the red blood cell. So now that we have this information, let's follow up with the question, what happens if we can't make NADPH in the red blood cell? Well, we know that NADPH is required to maintain levels of reduced glutathione. And we know that this glutathione is required to remove reactive oxygen species. Therefore, the loss of NADPH in the red blood cell could impact the lifespan of the red blood cell and cause hemolysis. Clinically, individuals with a deficiency of NADPH in the red blood cell, particularly due to deficiencies in glucose 6-phosphate dehydrogenase, often have clinical signs of jaundice, which indicates excessive red blood cell hemolysis. First,